For understanding the concept of electron transport chain, let us get back to the Krebs cycle, where there is formation of NADH and FADH2. Both act as hydrogen carriers in the process of electron transport chain. NAD positive is a hydrogen carrier derived from the vitamin niacin. It is a coenzyme involved in several oxidation reduction reactions catalyzed by dehydrogenases. In the example opposite, taken from Krebs cycle, malate dehydrogenase catalyzes the oxidation of malate to oxaloacetate. During this reaction, NAD positive is reduced to form NADH. If we talk about FADH2, FAD is a hydrogen carrier derived from the vitamin riboflavin. It differs from NAD positive in that it is covalently bound to its dehydrogenase enzyme and is therefore known as a prosthetic group. In the example opposite, the succinate dehydrogenase reaction is shown with FAD being reduced to FADH2. Succinate dehydrogenase is bound to the inner membrane of the mitochondrion and is an integral part of the respiratory chain. Let us talk about both of their electron transport systems separately. For NADH electrons, 3 complexes are involved that include complex 1, also known as NADH coenzyme Q reductase. The second one is coenzyme QH, 2-cytochrome C reductase, also known as complex 3. And complex 4 is of cytochrome C oxidase. Here complex 2 is missing, which is used for FADH, 2 electrons, so here we will only discuss these 3. Cytochrome C is attached to complex 3, which is responsible for shifting electron to complex 4. Let us talk about that later. Now here is matrix, and here is intermembrane space. There is maximum number of electrons in the matrix that will be going inside the intermembrane space to produce ATP from proton motive force. So here in the complex 1, NADH is oxidized to NAD positive and proton with the help of NADH reductase enzyme. Here 2 electrons are lost by NADH molecule. Now these electrons reduce FMN and iron sulfide complex that will form ubiquinol, and this ubiquinol will be reduced to ubiquinon. During this procedure, 4 protons are pumped inside the intermembrane space. Now this ubiquinon is also oxidized, releasing 2 electrons, and 2 protons are also pumped into the intermembrane space. Now these 2 electrons are accepted by iron sulfide, in which 2 more protons are pumped into intermembrane space. Now protons in iron sulfide complex are accepted by cytochrome C, and here the complex of cytochrome C carries 1 electron at a time and carries it to the complex 4. Remember, it will first carry 1 electron to complex 4, then come back to complex 3, and will again go back to complex 4 to give another electron. Now in the complex 4, cytochrome A is converted into cytochrome A3, while the 2 electrons left are taken by oxygen to form water molecule in the end. And in this phase, 2 more protons are pumped into intermembrane space. Now looking at the diagram here, you can see that 4 protons are pumped in complex 1. 4 protons are pumped into complex 3, and last 2 protons are pumped into complex 4 site. So from 1 NADH molecule, ETC will pump total of 10 protons into the intermembrane space. Now let us talk about FADH, 2 electrons now. Here, complex 2 is involved instead of complex 1, which is known as succinate coenzyme Q reductase. Now first succinate is converted into fumarate, which is also a step in Krebs cycle. 2 protons from fumarate will be given to FAD, and it will be converted into FADH2. Now FADH, 2 loses its protons to iron sulfide, which reduces to ubiquinol that get reduced to ubiquinon. In this process, 4 protons are pumped into intermembrane space in matrix. Ubiquinon ultimately gets back to the Q cycle. Further reactions are same as per we have discussed in electron transport of NADH. Now in this cycle, total of 6 protons are pumped into intermembrane space. 4 protons from complex 3 and 2 protons from complex 4. Now the total number of protons pumped from both of hydrogen carriers will be 16. 10 from NADH, and 6 from FADH2. So after the movement of protons comes oxidative phosphorylation, where there will be production of ATP. Now briefly discussing oxidative phosphorylation, in which will occur from complex 5, also known as ATP synthase. Now this complex has channel for backflow of protons. These protons will move back into the matrix producing ATP. So this is how oxidative phosphorylation is coupled with the electron transport chain. The details of oxidative phosphorylation will be provided in the next video, so do look out for that. Now remember that each ATP molecule is generated with 4 protons, and if we calculate the number of ATP produced by each carrier, then we will get the following equation. An ADH pumped 10 protons, so 10 divided by 4 will be generation of 2.5 ATP. FADH 2 pumped 6 protons, so 6 divided by 4 will be generation of 1.5 ATP. That was all about electron transport chain. Let us move towards some mitochondrial pathways related to it.